Hello, this is Mickey Urdea from Alteris Associates. Thanks for joining us in another episode of Interesting Stories in the History of Diagnostics. Today, I'm actually going to talk about my own work in the early days of HIV viral load. We didn't call it that then when it first came out, but that's what it became known as. But specifically, I want to talk about the value of measuring the quantity of virus in blood as an indication of the effectiveness of therapy. It starts back when I began at Chiron Corporation, one of the early biotech companies in Emeryville, California. That was a long time ago, 1981. And I had been a postdoc in San Francisco before that and joined Chiron very early. I started out in a building that was shared with Cetus Corporation. And Cetus Corporation, big claim to fame was the invention of PCR. And that was with Kerry Mullis. And we were actually in the same building. He was in a lab downstairs from my lab at Chiron. I started that laboratory to develop methods for gene synthesis. We actually made genes. One of the big genes that we made that was quite unique and successful was one for insulin. We made human insulin that could be cloned and produced in yeast. But after a few years of doing gene synthesis, I was looking for other things to do with that kind of nucleic acid chemistry. And at that time, people started to become quite interested in developing nucleic acid-based diagnostic tests. That became a real passion for me. I had a very specific idea about what to do with nucleic acid testing. I wanted to do quantitative measure of viral RNA. I felt like this was likely to have application in monitoring therapy over time, antiviral therapy. And fortunately, Chiron let me do it, which was wonderful. So the company GenProbe was one of the big pioneers in this field. And they had developed new nucleic acid-based tests for sexually transmitted diseases such as chlamydia and gonorrhea. They'd also been developing a test for TB. Those would be referred to as qualitative tests. That is, you can tell whether it's there or not. You can't tell how much of it there is. But I had this very specific idea in mind that I was able to pursue, which was being able to quantitate the amount of an organism in a sample. In particular, I wanted to be able to quantitate the amount of viruses in blood. I thought there was a potential that we could monitor the treatment of the viruses with antiviral drugs and see a change in the viral load. You won't believe this, but people thought that was a really stupid idea for a very long time. The first time I ever talked about it at a meeting, I was told by a very famous professor at Stanford that I just don't get why we need this. So I knew we had a long road ahead of us. But at that time, we were developing a technology that we called branch DNA signal amplification. And this was with my colleagues at Chiron, Thomas Horn, Brian Warner, Janice Kohlberg, Jenny Klein, Joyce Running, and others. We initially started working on hepatitis B virus, but Chiron was in the process of sequencing HIV, which was very big at that time in the late 80s. Everyone was working on it. Chiron was the first to actually clone and to sequence the virus in collaboration with Jay Levy at UC San Francisco. So we had the sequence. We knew what the sequence was. And we were working on this technology, and I thought this one should be great for us to do this work on quantitation of the amount of virus. So we worked on that for some time, and we developed the first quantitative method for detecting HIV viral RNA in blood.
And the first version of the test that we had a sensitivity of about 10,000 copies per ml of blood.
We didn't know how much virus there was in the blood. Nobody did. So we just went with it, and we started to do some work on it. And some of the people in the group, in particular, John Todd, was looking for opportunities to get samples for HIV. And John made connections with John Mellors at the University of Pittsburgh. John Mellors had a set of samples from what was called the MAX cohort. And these were samples provided by gay men who had high risk for HIV infection and followed them over time. And there were some people that rapidly progressed towards AIDS and some that did not. They were much slower.
And John sent us a set of samples, a portion of which were for men who had developed AIDS rapidly, and another set for men who did not.
And we tested them.
And what we found was that the men who did not progress rapidly towards AIDS had a very low viral load, below 10,000 copies per mil. And the men who progressed rapidly to AIDS actually had a much higher viral load.
That was kind of amazing. We were very surprised by that. And John Mellors was really surprised. Afterwards, we developed a publication with John Mellors that was published in Science about the importance of viral load differences in people that progress rapidly versus slowly to AIDS. Actually found that the amount of virus in the blood was actually directly related to whether or not someone would develop HIV disease, in particular AIDS. So we started working with others in this area as well, one of whom was David Ho at Aaron Diamond Research Center.
It was introduced to us by our chief medical officer at Chiron, David Chernoff, who actually played a significant role in this whole story in HIV viral load for several years. And then we began testing samples for David Ho with our new technology at Chiron.
And he had samples where people were put on this experimental drug for HIV treatment, AZT, and he found that they had sort of their personal level of RNA using our technique that's rather stable. Give them the drug, it drops. And then when they took the drug away, it popped right back to where it had been before. It was very clear using the viral load test. That sort of just opened the floodgates. People started getting very interested in using this. We had a lot of collaborations with people testing this, and it became important to use this in clinical studies where people were looking at new drugs. The protease inhibitors came out later. They dropped the viral load even further, combination drugs, all those things just really took off. Roche came out with a comparable technology to ours. And in fact, theirs was more sensitive than ours, using PCR. In fact, I'm pretty sure that they started working on quantitative PCR because we had shown that quantitation was so important. And before that time, PCR was just qualitative. They did a really nice job. It was a very sensitive assay. And I had to reorganize the team at Chiron to equal them. So we spent 10 months very quickly, put a new product out, which had comparable sensitivity, almost identical to Roche. And we just all fought it out in the field. And it's good to have good competition. It stimulates both sides, and they worked out very nicely. I worked at Chiron for 17 years on this. We spent a lot of time trying to introduce and create the market for HIV viral load. Roche as well, other competitors came along. And to this day, the market for HIV viral load continues to expand, which just amazes me. It's going into many other countries. It's in most countries now, increasing the number of people being tested to monitor the effectiveness of their drug treatment. And then after that, we started working on HCV, and we had an HBV test as well. So that general concept held up. And it went from a situation where people were doing clinical trials based on outcomes, HIV-infected individuals moving toward AIDS, to showing that the so-called surrogate marker, viral load, if it dropped, that was an indication, and this took time to prove, that the person was going to be well for a long period of time as long as the viral load was suppressed. That's still what we do today. So that was a long time ago. It's been quite an interesting ride since then, and glad to share a little bit of the early days of HIV viral load.
During the telling of this story, I mentioned a number of people that were key parts of the team at Chiron, but I want to just mention the fact that I didn't get a chance to name even half. It was a large group of people that participated in this and made major contributions over a long period of time. And I thank everybody that was involved.
Thanks so much for listening to another episode of Interesting Stories in the History of Diagnostics. This is Mickey Urdea. Thank you very much. Bye.